that was a very nice introduction, and I thank you very much for that. Basically, my career, I started off in veterinary science, but I went into that with the idea of doing research on diseases of domestic animals and trying to increase food production, because at that stage, way back then, we were very concerned about feeding the world. We're still concerned about feeding the world, though we think a lot more in terms of plants than animals at the moment. And very at the beginning of my career, I started doing some research on a bacterial disease called Leptospirosis, which infects us and also infects cattle and dogs, and it grows in the kidney. And I became very interested in, as that infection resolved, as the animal cured, lots of white blood cells from the blood, came flooding into the region where the bacteria were growing and were clearly involved in eliminating them. This is what we call inflammation. Cells coming in from the blood to a site where some sort of infectious organism is doing damage. That was a bacterium, a spirochete, quite a fascinating thing. And then when I went to Edinburgh, I worked in a place that worked on diseases of sheep. And I started to work on a disease called Lalpineal Encephalitis. This is a tick-borne encephalitis virus, a virus that's transmitted to us by ticks, biting ticks, or to sheep, really, much more than to humans. And equivalents are Russian spring-summer encephalitis and various infections with names like that. And I became very interested in what happened in the brain. So I became a brain pathologist as well as an infectious disease person. And I again became very interested in the cells that came out of the blood and started flooding into the brain. And that's really what my career has been about, finding out how that cellular component in recovery from an infection actually works. And I focused on one part of that. Now, I could have gone in one or two different directions at that stage. I was fascinated with neuroscience, and I was in contact with a lot of neuroscientists. But instead, as it turned out, I went down the road of infection and immunity. But I've always been interested in that relationship between the brain and the immune system. And this works in very, very many complex ways. Now, talking about COVID, of course, we know that the brain, the sensory apparatus, I mean, the sense of smell initially, can be very much affected. And we know that people, after the initial phase of infection, when they think they're getting better, can come down with, well, brain symptoms of brain fogs, and we call this long COVID. So I'm going to talk a bit about that and talk a bit about brain, a bit about immunity in a very, very, not great depth, but try and give you some basic ideas. Now, as human beings, we're large, complex, multi-organ, multicellular systems, and we have to be able to defend ourselves against totally novel infectious agents that are tiny. I mean, you could see the
recording in progress
of leptospirosis down a microscope, but on a light microscope, you don't see the sort of viruses that worry us in humans. You can see them down an electron microscope. So these are very tiny organisms. They've got little bits of DNA or RNA. They multiply very rapidly, much more rapidly than we do. But we have to have a very sophisticated immune system, and a very specific one, to keep them at bay and stop them from overtaking us. And as we know, we're constantly challenged, and we have been for thousands of years, more so since we started moving into communities where we got a lot of people together, with pathogens, infections, viruses, bacteria, fungi, and so forth, that come out of nature. And they often come out of wildlife. And of course, that's the situation with COVID-19. It's caused by a virus called a coronavirus, which has undoubtedly come out of some bat species into us. The bats, it turns out, gosh, not getting us to move on. Here we are. Right. Bats, it turns out, have an immune system that operates rather differently from ours. Like us, they're mammals, and like us, they're large and complex when you think of them in terms of a virus. But their immune system works a bit differently. It's got the same basic themes, but through evolution, the adaptive immune system, which is the part of the immune system which is very specific, has evolved over 350 million years, in fact. We trace it back to the bony fishes in the ocean, and it's over those 350 million years. And as it's gone into different species, it works a bit differently, but it achieves the same thing. The mammalian immune system is a bit different from the avian and the reptilian, which is more on the dinosaur lineage, we think. But of course, mammalian immune system, bats are mammals, we are mammals, and so it has similarities but differences. And it seems that bats can tolerate carrying a lot of different infectious viruses, not actually get rid of them, but just live with them. And that makes them a very big risk for us, because some of these viruses have the potential to jump over into us, often via some intermediate species. We knew that happened in 2001, 2002, with the original SARS virus that killed about 800 people in Asia and was very lethal, but much more lethal than this one, but didn't spread nearly as much. And we know that that came in through a little animal called a civet cat, which was sold in live animal markets in China. China has a practice of selling live wildlife that is then used for human consumption. And it's these wildlife, these live animal markets where people and wild-caught animals come together, including bats, that are particularly dangerous for these viruses to jump across into us. Now, there are loads of these coronaviruses in bats. Before the year 2000, we knew of 2 of them circulating in humans. They caused common colds and Krupp-type syndromes. But since 2000, 5 more have jumped into us, 3 of which cause serious disease and 2 of which are common cold coronaviruses. So what's changed in the world since the year 2000? What's changed is that China has become much more prosperous, and there are many, many more people traveling to and from out of China on airplanes. And it's airplanes that get these viruses around the world. So as we return to normal in international travel, these viruses will continue to be a considerable threat. Now, it's another talk completely about what we do about that. There are things we can do, and I think we have to move very aggressively towards that. They also carry some other viruses which are really dangerous. Another family called the Hanover Viruses that have caused problem in Southeast Asia particularly, and filoviruses, the Marburg, Ebola type virus, and most of you will have heard of Ebola, which caused horrible, horrible hemorrhagic disease. So bats, we don't want to kill all the bats. They're very important species in nature. They get seeds around, they eat insects, they do many important things, but bats are a real threat, and bringing bats and people together is, as far as we can avoid it, not a really good idea. Now, this is the coronavirus. It's a tiny thing. It's got a genome of about 30,000 base pairs around, compared with the 3 billion base pairs in our genome. But as you know, that tiny virus has taken over our global economic system, changed the way we live, and of course it's killed quite a lot of people. So these things are really dangerous, and we have to be able to control them and get rid of them, and they're coming at us all the time, different types. So, and viruses are what we call obligate intracellular parasites. These are the most intimate of the infections. The virus doesn't have any way of moving itself around. It only moves in air or in water or whatever it survives in. And it can only multiply and divide when it gets into living cells. So what coronavirus does, the SARS-CoV-2 does, is hijack our cells, gets into them, takes over some of our cell machinery, uses its own genes to then make new viruses. In an evolutionary sense, all viruses want to do is survive. So all they want to do is multiply and infect other people or whoever, whatever species is transmitting them. So it's a very simple life cycle, if you like, but it's also one that we absolutely have to interrupt unless we're going to have severe problems. If you can't get rid of the virus, then it will keep infecting other cells. So part of the immune system does that. It gets rid of the virus in free form. If you can't get rid of the virus-infected cell, they will keep producing cells. And with this virus, it can go cell to cell. It doesn't have to necessarily go outside and infect other cells. So we have to get rid of the infected cells. And I'll tell you a little bit about both those things. So to deal with infection, we have this complex adaptive immune system. We also have another arm to the immune system called the innate immune system. That's a very rapid response system. And some of the toxic molecules that are produced by that innate immune system can actually make us feel pretty sick. That's the immediate response. The fever you get, for instance, can be due to some of the cytokines like interleukin-1 that's secreted from some of the cells of the innate immune system responding to a pathogen. Fever and being drowsy, which can be effects of these cytokines, are nature's way of telling us to slow down so we don't pump more virus around our blood, for instance. So the word immunity itself comes from the Roman soldiers in the Roman state of ancient Rome, who after they'd been off in Europe, maybe beating you guys up, went back to Rome, and then they were for a time immune from tax. The veterans didn't pay any tax. And so what immunity is about is defeating the tax of infection and overcoming that. That's where it actually comes from. And immunity is there to constantly deal with novel challenges. Now, just to digress a little bit, I started in neuroscience as well as immunity. Humans have two great sensing and responding systems that allow us to deal specifically with external threats. And we're celebrating on this meeting and this occasion, we're celebrating the neuroscientist, great neuroscientist, Bert Sakmann. So Bert comes from the neuro side, neurology, neuroscience. And his study is to do with brain and signaling and all those things. And of course, he's known for the development of a technique called patch clamping, which is a really important technique in cell biology. We use it in immunity as well, because it's down to the level of the cell. But his basic focus has been on the brain and the neurosystem. Alongside that, though, we also have this other great sensing and responding system, which is the immune system. And they're very different. Once you go beyond the molecular mechanisms, obviously you can think of many differences just offhand. The immune system operates below the level of consciousness. It can impact on consciousness, it can make us drowsy, make us feel sick, but we can't think our way through to get a better immune response, not as far as we know at least. And whereas the nervous system is both at the level of consciousness and below the level of consciousness, and it's very complex. And we use the same words. We use words like recognition, response, recall, memory, but they mean actually very different things. The nervous system's hardwired. It's anatomically stable. Your brain doesn't move around. It grows at a sort of constant rate, and it stays the same size, hopefully. But the immune system is mobile. It's moving around the body. It's flexible in size. It's dispersed. It's in constant motion. Sometimes it's sitting in places, and we don't know a lot of the details because it's very, very difficult to actually study the immune system in the individual, particularly in humans. You can do a lot more in mice, obviously. And the immune system, whereas the brain works by recognitions and circuits and all this sort of thing, the immune system works by cell division and replication. And the cells of the immune system, they are both the sensing element through their specific receptors and the responding element through the various effector functions like making antibody or being killer cells. The nervous system has a central processing unit, the brain. The immune system has no central processing unit. The cells of the immune system are totally independent. They do interact at times to promote immunity, but they're totally independent of each other, and they don't, in a sense, know each other. So there's the nervous system. We're all familiar with it. What response means, recognition, some receptor recognition by eyes or touch or something, then signals that go out. and they're transmitted by nerve cells to the brain. Signals that go out by other nerve cells to operate motor neurons, which trigger the muscles, and that's the effector arm of it. But with the immune system, the effector arm are the cells themselves and the clonal lineages of white blood cells. So two great sensing and responding systems. Of course, we divide them up because of the interest of academic disciplines, so we can study them. But of course, the body doesn't. And if you want to have this type of thinking, if you want to think of yourself in this way, and you're interested in, say, university training and so forth, obviously medicine's one way into this, because medicine looks at the holistic, the way the various systems that we talk about interact. Now, the basis of vaccination, which is our main protection against infections, is immune memory. And immune memory works by the persistence of what we call memory cells. These have started out as what we call naive cells. They've then differentiated in response to the infection into the antigens. So these are the proteins and so forth on the virus. And then some of those cells have come out the other end and become memory cells. Some others, and which float around, they can be around for 50 years, those lineages of memory cells. Others become what we call the effector cells, the cells that actually do the job of getting rid of the pathogen. Now, we didn't know much about this, actually. We knew about antibodies and we knew about immunity from the turn of the century. And really, the first Nobel Prize for medicine was for antibody therapy. But we didn't really know about the cells of the immune system until the 1950s, when they were really discovered their function. They were known. They were the most uninteresting cells in the blood, according to the hematologists, the people who look at blood, the lymphocytes. The lymphocytes are small. They don't have much cytoplasm, and they're just boring-looking cells. And so the hematologists didn't like them because they had wonderful cells they could stain up to look fantastic that they call eosinophils and basophils, and they look really great when you see them stained down a microscope. Lymphocytes really boring, but they're the most important cells. They're the cells of adaptive immunity. We knew before the lymphocytes were worked out that some cells pump out these antibody molecules into blood. This was discovered actually by Astrid Fagrius working in Copenhagen. And they're called plasma cells. And that's an anatomical decision. They're big cells with lots of cytoplasm and lots of organelles and so forth in that cytoplasm that are making a lot of protein. And then Jacques Miller and various people, like Henry Claim and various other people through the 60s and 70s actually showed us that these lymphocytes divide into two substantial lineages, the T cells, which are processed through the thymus up in the neck, and the B cells, which are the precursors of the antibody-producing cells, and the plasma cells that make the antibodies. In the chicken, there's a specific organ that does that. It's called the bursa fabricius. In us, there's not. These cells come out of the bone marrow. So this is what an immune response looks like. We start with maybe a very rare cell with a very specific receptor. They go on in the lymph nodes, the glands that swell up in our neck or that swell up in our armpit, perhaps, and are painful after a vaccination. That's where the immune response happens, in the lymph nodes. These are kind of drainage stations on what we call the lymphatic system. The lymph is the drainage system of the body. We have the blood, which is pumped around the body by the heart, and that provides all the oxygen, all the rest of it. Then fluid that goes out into tissue comes back into the blood through the lymphatics. The lymphatics don't have their own pump. We pump it with our muscles. That's why when you're sitting on a plane for hours, you'll get your legs may swell because of the accumulation of lymph, because you're not pumping it through motion. And the lymph nodes are these filter stations along the lymphatic system. That's where immune responses happen. That's where the very few cells with a specific receptor, say for the spike protein of the coronavirus, come in. They recognize it, and then they start to divide. And as they divide, they differentiate. And they can differentiate down slightly different lineages. Then we make our antibody-forming cells, our plasmablasts, the precursors of the antibody-forming cells, go out into the blood. The killer T cells that go out and wipe out virus-infected cells. And then when that all dies down, a lot of those cells may die off because you've got this enormous expansion in cell numbers. And then you'll get more cells, though, sticking around as memory cells. Now, we couldn't have that going on in the brain. If every time you had a thought, cells multiplied in your brain, you would have terrible headaches because the brain can't expand. It's encased in the skull, and brain swelling can kill you. So the immune system is happening out in cells like this. Of course, as the lymph nodes swell, due to the immune response going on, there are a lot of multiplication. You may get some pain because the nerve endings in the capsule of the lymph node and the receptors there may give you some sense of distension and pain. Now, adaptive immunity works primarily, not totally, but primarily by protein-protein interactions. Now, as I said, the plasma cells are cells that pump out lots of these antibody molecules. And this is what most vaccines do. They make antibody responses, and that's what vaccinologists and immunologists have been measuring for a very long time because they can be measured in the blood. And so the antibody molecules, this is a molecule called IgG. They're pumped out. They have this antibody binding site, and they tend to recognize folded components on proteins, what we call tertiary structure. And you can look at antibody. This is COVID-specific antibody responses induced by vaccination. You can look at the numbers of plasmablasts, B cells, that are produced by vaccination, produced by infection. You can look at booster effects by the increase in antibody levels or the increase in the number of antigen-specific B cells. This is from Ali Elabedi, who's at Washington University of St. Louis. And so you can quantify all this. You can actually quantitate what's happening. And of course, if you want to do good science, you have to be able to quantitate, measure, get numbers. And a lot of those capacities have only really developed over the last 30 years, apart from measuring antibody levels. But looking at the cells, it took a long time to do that. And this is how the protective antibodies work. I mean, here you have the ACE2 receptor on the surface of one of our cells. It could be in our nose, it could be in our blood vessels, it could be in our heart or our kidneys. And by binding to that ACE2 molecule, the spike protein of the coronavirus gets entry into the cell. I won't go into the mechanism, but there's kind of a molecular machinery there that does that. Now, if you've got an antibody molecule which can interpose itself in there, just like that, then that can't happen. So the ACE2 molecules can be blocked from the infection getting into the cell. Now, of course, if you're thinking about a coronavirus coming into the nose, it has no motor to move it around. If you think about antibodies in the nose, which after a vaccination are just spilling over in blood, there's a kind of a dating game going on. And if the virus hits the cell with the ACE2 molecule on first, it's going to start infecting cells. And in actual fact, even in very well vaccinated people, the virus can often get into cells before an antibody molecule will pick it up and stop it getting in. And as we know with Omicron, the antibody response is not that good anyway, and we're getting a lot of reinfection, and we're getting infection of vaccinated people. And that's all perfectly because the vaccine no longer matches so well, and the antibodies are against the previous virus. Though what does seem to be working is that other half of the immune response, the T cell response, which is knocking out virus-infected cells, which is why even if people get infected with Omicron, they've got much less chance of ending up in hospital and being very sick if they're actually vaccinated. But it's all probabilities, and you have to think in those sort of terms if you to understand this type of biology. Now, the other component of the immune system is the killer T cells, the one that I've studied. They're more like the individual Roman soldier. The Roman soldier got up close. I mean, they did throw a spear called a pillium, and that might be more like the antibody molecule, which is floating around in the blood and can float around for years and will be constantly renewed from the plasma cells. But the actual killer T cell has to get up close and personal. It has to make contact with the virus-infected cell. And so it's like the gladiator. The gladiator actually had a very, very short stabbing sword. They didn't have big, long swords they waved around, and that was their great strength. So this is a killer T cell. Here's a killer T cell, and it's going to kill this cell, which is virus-infected. And now it's stopped. Now, there we go. It's going to kill it. Now, watch this. It's in pink stuff, so that means the membrane has disintegrated and the pink stuff flooding into the cell. And you can see it's killed that cell. As it kills the cell, it kills the virus. It's a process we call apoptosis. And actually, it's the mechanism cells use to commit suicide. This is the recognition event. This is a specific T cell receptor. What it's recognizing is a little bit of the virus, a bit of peptide, tiny bit, 8, 12 amino acids. They're presented in what we call the transplant molecules on the cell surface. This is the stuff we got the Nobel Prize for. It's complicated. I'm not going to try and go into any detail, but we've all got these transplantation molecules on the surface of our cells. They're very, what we call very polymorphic, and they present these viral peptides in their tip. And that tiny bit of virus, that little peptide of 8 amino acids presented in that molecule on the surface of a cell, is what makes the cell look different, and the killer T cell will destroy it. Now, it won't destroy the cell next door, which has those same transplant molecules, but doesn't have that bit of viral peptide. So it's a very specific and targeted killing. You don't want cells going around the body just killing indiscriminately. And this is an immune response. This is actually from an influenza virus in the nose. It's actually in a mouse. And what we're looking at is the cells, the killer T cells, coming flooding into the fluid we can get back from the lung. We call it bronchoalveolar lavage. And this is what we call a primary response. It's a mouse, never been infected before. And here we see the immune cells coming in at about day 7, building up. The actual virus is eliminated from the lung about here. And these mice have actually been primed up. They've got immunological memory. But you can see, though the immune response is much bigger after what we call a secondary challenge, the recall response, memory recall, you see it's not there right away. So the T cells always take a time to get going again. They're not permanently activated, which is why if you're infected with Omicron, you don't have enough antibody, you'll still get infected. But it's these T cells turning on quicker, which wipe out the virus-infected cells and start you on the pathway down to recovery. And we can trace the lineages of these things. We can trace their receptors. We can do very sophisticated science. This is my colleague, Paul Thomas from St. Jude. He's just published an absolutely wonderful paper on this, all done in humans. One of the things that COVID has done has allowed us to do an enormous amount of immunology in infected people that we were never funded or focused to do before. But now we have a much better understanding of aspects of the immune system than we had before. Are the T cells, these killer T cells, protecting humans? Yeah, there's pretty reasonable evidence. There is. It takes a time to recall them, so they're not there straight away. Then people tried to make HIV/AIDS vaccines that worked against the killer T cell. Didn't work, never could work, because the AIDS virus comes in, it's got a reverse transcriptase, copies back into the genome before any of that happens. But with COVID and with flu and with a normal infection, yes, they do it. They can do the job of providing a reasonable level of protection. May not stop you getting a bit sick, but it may stop you getting very sick, and that's kind of important. Being a little bit sick is a lot better than being very sick. So here are some of these cells from COVID. I'm not going to go into details on this, and we can induce them with the mRNA vaccines or with the DNA vaccine, the adenovirus vectored vaccines and all the rest of it. So we can prime this component of the response. But of course, we're only priming it against peptides from the spike protein, which is all that's in the common vaccines, other than the killed Chinese vaccine, for instance. And so there's a lot of all the other proteins in the virus, when you're infected, can provide peptides that stimulate that killer T cell response. So if you've been vaccinated and then infected, we're sort of hoping a lot of people are super immune because they will have a better killer T cell response, or a broader killer T cell response. And the vaccine will have helped a lot with the antibody. So just to say a bit about the disease, when we first saw COVID, we were thinking of it just pretty much as a respiratory disease. The people who do imaging were telling us the lungs look horrible. I mean, they've got these what they call ground glass lesions, which looks hideous. Fortunately, they seem to repair reasonably, but it looked really awful. But there's a difference, and we were saying, well, this isn't influenza because it's a different virus, and we know they've got different properties. But we were thinking of COVID a lot like influenza, and the key thing that distinguishes influenza from COVID is the influenza viruses, which we've lived with for a very long time, of course, don't normally get into the blood and go around the body in the blood. They're not what we call viremic. They'll do that in birds, but they don't normally do it in humans. It's very unusual to find influenza virus in the blood in humans. But COVID, the virus gets in the blood, and it's what we call a systemic infection. Now, of course, once the virus is in the blood, it's in the superhighway of the body. It can go anywhere in the body, and the ACE2 receptors that allow it to get into cells are right through the body. They're in the heart, they're in the blood vessel walls, they're in the kidneys, they're in various places. And a lot of the people who are severely affected by COVID are, for instance, diabetics, whose kidneys are not working all that well anyway. Then you get an infection with the coronavirus on top of it, and they're in very bad shape. And apart from that, you can see it in flu, in late-stage flu, you can see some clotting problems. But this virus induces major clotting problems. And it's not totally unusual if people die acutely and unexpectedly. They're actually dying from a stroke or a heart attack due to blood clots. The terminal alveoli in the lung where the gas exchange occurs, here we see it, can be loaded with these microclots, which is disastrous. And once the doctors understood that, they started giving low-dose anticoagulant like heparin fairly early on when people were hospitalized. That saved a lot of lives, as did anti-inflammatory drugs, very simple ones like dexamethasone. So that's the acute phase of COVID-19, but there's another element to it which is very concerning, and that is this long COVID phenomenon, where people who've had a very, very mild early clinical course can develop this persistent debility. We've seen this with viruses before. We've often not known what the trigger is. It's been called myalgic encephalomyelitis or chronic fatigue syndrome. And the long COVID has many elements in common with that. And of course, the difference between that, and it can infect young children, though that usually not for as long, but people can be very debilitated. And in some cases, they get better. And in some cases, it becomes progressive. This is a very real concern that's going to cause it's causing a lot of human suffering and may actually have a lot of impact on productivity in societies and all the rest of it. So we really need to understand it better. What we have with COVID is a situation where we have the initial diagnosis. In a country like Australia, early on at least, we're now using rapid antigen tests. Everyone who had any symptoms, or often people who had no symptoms, were tested positive by a PCR test. Very, very sensitive test. And basically, that means with these cases of long COVID, we've got some long-term, what we call clinical cohorts on clinical trial, where people have been examined since they developed mild symptoms, then they went on to develop or not develop long COVID. And we've got people looking at what's going on within them to try and understand what's happening here. And it's an interface. There could be various mechanisms at work, but part of it could be due to inflammatory cells producing these cytokines and chemokines going into the brain. Part of it may be due to the accumulation of molecules that can be associated with inflammation in the brain, molecules that the neuroscientists think a lot about when they're thinking about Alzheimer's disease and those diseases, molecules like amyloid and tau proteins. And you can find these proteins at high level, say, in people who actually do die from COVID. So there's a whole issue and a whole interface between immunity and neuroscience that will need to be unpicked here. And I think it's going to take a while to do that, but I think we're in better case to actually do it. There may also be what we call an autoimmune component. The problem with our immune system is it's there to deal with what the foreign, the dangerous coming in, but it can sometimes turn around because of cross-reactivity sometimes. A protein on a virus that cross-reacts with a protein in us, it can turn around and start to attack ourselves. These are the autoimmune diseases like rheumatoid arthritis, multiple sclerosis, type 1 diabetes probably has that in it as well. And there's a number of these diseases. And again, we've suspected viruses as triggers for a long time. But having that specific diagnosis with COVID may actually show us whether these particular viruses are triggering some of those problems. So there's a lot of science to be done, a lot of medicine to be done, and a lot of research to be done to really understand these diseases. But I think we're in a great position to really start to move some areas of medicine forward, particularly perhaps neuropathology, CNS damage caused by viruses, and the chronic sequelae, and also elements of basically brain pathology. So that's it. That's where I'll stop. And thank you. And I hope you're having a good time and it's not too early in the morning for you.
Dear Peter, thank you very much for your really fascinating presentation. So that's definitely somebody didn't wake up before the presentation. Definitely your presentation did that. So ladies and gentlemen, the stage is yours, and we are looking for questions.
Do you think that because COVID has so many mutations, it's going to remain in society, or is it going to disappear at some time, or just live with us forever?
Well, we would love to think it will disappear, but most of us think it probably won't. What we would like to think is that what's happening with Omicron will be the beginning of it going to a milder stage. But there's absolutely no guarantee that a more pathogenic mutant may not come back. And so it's really quite concerning because though early on, for instance, we shut down Australia and kept the virus out for quite a long time, there's no enthusiasm for doing that anymore. And currently with Omicron, we're getting 40,000 plus cases a day, with very low hospitalisation rates, because our most vulnerable population, the elderly, have had they've all had at least 2 doses of vaccine. Many have had 3, and some are now having 4. I've just had my 4th. I'm 81 years old, so I'm quite keen to have it. So the vaccine, by keeping the virus out till the vaccines became readily available, and they did very well up to Delta. The vaccine was protecting very well against Delta early on, not quite as well against Omicron. There are Omicron vaccines in trial. They could be out there quickly if they're shown to be a benefit. But it may be that we're through Omicron by the time this is over. So it would be great to think it'll go away. No guarantee it will. But we'll be in a much better shape to handle it. And the reason for that is we know how to make vaccines against it. And we can make them very quickly. The only reason the Omicron vaccines aren't out there is because they haven't been shown to be that efficacious so far. And but they don't need to be tested through the enormous vaccine trials that were done early on. Each trial may only involve a couple of thousand participants before, say, the American FDA registers the vaccine. And the other thing, though, which I think is more important in a way, is we're now starting to develop specific antiviral drugs that are specific to the virus. But we can develop antiviral drugs that are specific for the whole class of viruses. We've got, we control AIDS, HIV, with drugs. People get a cocktail, 2-3 drugs a day or a week, depending on how long-lasting the drug is. You need to have more than one drug that works by a different mechanism. Otherwise, the virus can mutate towards a resistant form. But if you've got 2 or 3 different mechanisms in virus replication targeted by the drug, then you can stop these things. So we've now got one drug that looks pretty good called Paxlovid, another one called Molnupiravir, probably not as good. These drugs are in short supply, but they've been registered. And if we could, imagine this. Imagine a new coronavirus gets in, and we've got, say, either a PCR test set up for it, or now we might actually have one of these rapid antigen tests, these lateral flow tests that people can test at home. Say that you felt off, you knew this virus was around, you did one of these rapid antigen tests, and you read after the 15 minutes or something, and they cost a few dollars, but they're not that expensive. And you read the antigen test, that tells you you've got the virus. Then you report it back to the website in your particular country, the health website, and that automatically activates a brief review of what they know about your medical history. And if you've got a national health system, they'll know quite a bit. A brief review, and then basically then a drone pops up, or a guy on a motorcycle that normally delivers pizzas or Chinese food comes to your door, gives you a packet of 5 pills, 5 days worth of pills. You take those for 5 days and it knocks out the virus. So that may be where we can actually protect ourselves. And there's a big international effort now, really driven very hard by Tony Fauci in the United States, but elsewhere as well, to actually develop these panels of drugs that can be used against all these dangerous pathogens. We've got them for flu. They're usually not very good because you never use them early enough. But if you use the approach I've just described, where as soon as you got a sniffle, you did a test, as soon as you're positive, you get some pills, we could probably turn off most of the flu. So basically, within 5 or 10 years, I think we're going to have a whole spectrum of drugs that could be used to tackle, say, any new virus that comes out of nature. Of course, drugs are expensive. Vaccines are a lot cheaper, and we'd prefer to use vaccines, but having the drugs there would be a great backup.
Peter, one question. What can we learn from the bat? The bats tolerate pretty well the coronaviruses. What is their trick?
I mean, as you imagine, the number of bat immunologists on the planet are pretty small. There are more of them. The great Linfa Wang, who worked for many years in Australia and did a lot on the Hanover Viruses and on corona, now in Singapore at the Duke University campus in Singapore, has been focusing on that. We need to learn a lot more about the bat immune system and how it works. And we think it's something to do with the interferons and how, and I know there's evidence coming through on this. But of course, you know, it's only recently that anyone on the planet who controls medical research funding would be interested in funding research on the immune system of the bat. But I think we'll understand this a lot better, and maybe understand a lot better how these species live with the viruses in the way they do.
Thanks. So my question will be, can we predict how a virus will mutate, or it's totally random and there's no algorithm?
No, unfortunately, there's been a lot of effort put into trying to do predictive models and trying to predict how a virus will mutate. All the major lineages we've seen with this virus have just selected themselves in nature, and basically they're not related. So with Omicron, we have the variants. We have BA1, BA2, but Delta, Omicron, Alpha, Beta, and so forth have all come from different mutational events, and they all seem to be quite separate. Now, maybe we might settle into a pattern where we're dealing just with one lineage of mutants. You see, what's extraordinary about COVID, which makes it different from influenza, there's a lot of background immunity. The T-cell responses are very cross-reactive between different flu viruses, even different pandemic strains. But we had none of that, well, very little of it with the coronaviruses. And so, but now we're growing to a situation where the whole planet is getting to a stage where they have some immunity. And they also either have it because they've been infected or because they've been vaccinated, or both. So we think it'll be very different. What we had initially was what's called a virgin soil pandemic, where nobody had really experienced these things before at any high frequency or any degree of severity. And now we've had a totally different experience. We do know that the people who survived that very early SARS coronavirus, which is quite a bit different from the one we've got now, they do have a good measure of protection. They've had a good measure of protection against CoV-2. And we also know some of the monoclonal antibodies that were made. I didn't get to talk about those, but you can make single lineages of antibody, and they're used in therapeutics quite extensively. Some of the monoclonal antibodies that we've used against CoV-2 were actually made against that original corona. They're cross-reactive, and they cross-react between different mutations.
I would like to ask if this recognition that the T cells do does also work with the prions or only with viruses?
No, prions. I know my colleague Zinkernagel did a bit on this, but there's not much been done on it. Prions are, these replicating proteins that make copies, and they don't seem to generate much in the way of immune response. Now, I did look at this years back, but there's no real evidence of solid immunity. And a prion disease, we call them Spongiform Encephalopathies, like Creutzfeldt-Jakob disease or Scrapian sheep, which is the prototype, or mink encephalopathy, or the other one, of course, is Bovine Bse that infected humans. They don't cause much in the way of inflammation. They don't drag immune cells in. But what they are associated with is accumulations of amyloid and things like tau proteins, and with vacuolation of the brain in histological sections, which you can see a bit in COVID. So maybe there's something will come together there, but basically the prions don't generate significant immunity. Now, someone may correct me on that, but I think that's where we still stand.
So if there is no more question, we would like to really, really thank you very much for your time. Last time you also were with our students, and of course, as usual, concerning this conference, again, I would like to actually show you. I hope that the camera will change, and you will be able to see that we will have, of course, the regular medal for the conference, and all of the people who could personally attend signed for you, and thank your effort about being with us also today. So thank you very much, and we wish you all the best.
Well, thank you very much, and I wish you all the best for the rest of your meeting. And please pass on my very best regards to Bert Sakmann. So, bye.